Good day, and thank you for standing by. Welcome to the Prometheus Biosciences Phase 2 Data Readout Call. At this time, all participants are in a listen-only mode. After the speaker's presentation, there will be a question-and-answer session. To ask a question during the session, you'll need to press star 11 on your telephone. Please be advised that today's conference is being recorded. I would now like to hand the conference over to Chairman and CEO, Mark McKenna. Please go ahead.
Thank you so much. Welcome, everyone. Good morning, good evening, good afternoon. Thank you for joining us for our Phase 2 data release. But first, we'd like to remind you that today's call contains forward-looking statements, including remarks about our plans and expectations, as noted on the adjacent slide. Please review our latest 10-Q filing for additional information about the risks and uncertainties inherent in our business. During the presentation, our Chief Medical Officer, Dr. Allison Liu, will describe the details of our Phase 2 study results, followed by our Chief Scientific Officer, Dr. Olivier Laurent, who will discuss the CDX interim results, as well as our translational data that we've collected so far. We'll conclude the call with Q&A, which will include broader management, as well as our KOL and head of our Scientific Advisory Board, Dr. Bruce Sands. Today is a momentous day for patients suffering with IBD. As many of you know, the efficacy ceiling in IBD has been stagnant for decades, with very few, if any, agents surpassing the 10 to 15% placebo-adjusted clinical remission efficacy threshold. In addition to underwhelming efficacy, many approved agents have shown safety and tolerability issues. These result in IBD patients seeing static results in the rates of surgery and Crohn's disease over the past couple of decades, and regular cycling on and off of available therapies, hoping to find something that works. While we see new product options, including orals and topicals, these therapeutic options, unfortunately, can sacrifice efficacy and result in safety issues for patients. What the field has been looking for is an agent with disruptive efficacy and favorable safety that can be more meaningful, have a more meaningful impact on patients. 20 years ago, Prometheus Biosciences founders Dr. Targan and Dr. McGovern set out on a mission, along with the late Dr. Tachi Yamada, to address the unmet need in IBD and other immune-mediated diseases. Prometheus was established to address the shortcomings of existing therapies offered and brought a new approach to drug development in I&I, one of the largest therapeutic categories in medicine. Their vision was to incorporate genetically associated targets that have a greater probability of success in the clinic with biomarkers to identify patients most likely to respond to therapies. Today is a groundbreaking event, not only for Prometheus Biosciences, but for the field of anti-inflammatory and fibrotic diseases. We're announcing a clinical candidate that delivers best-in-category efficacy and a clean safety profile. This agent was born out of a strong understanding of TL1A biology and through a one-of-a-kind data set built by our founders that provide clinical metadata, genetics, and samples. This clinical genetic database has provided us a pathway to precision medicine in IBD and other immune-mediated diseases. Before we jump into the data, let me provide three major outcomes of our Phase 2 studies in UC and Crohn's disease. First, a new high watermark for clinical performance showing differentiated combination of high efficacy and favorable safety in IBD. Importantly, this was achieved despite the very severe patient population. Second, the first precision medicine result in I&I showing a positive trend towards selection of high responders using a companion diagnostic. Based upon our confidence in the data and interest in moving quickly, we conducted an interim analysis of cohort 1 to assess our companion diagnostic and saw early but very promising results. And third, a pleiotropic drug showing both anti-inflammatory and anti-fibrotic clinical activity, paving the way for new indication expansion, MOA differentiation, and additional confidence in our SSc-ILD program. We believe these data fully support advancing PRA023 into registrational studies in both UC and Crohn's. In the TL1A space, we are the clear leaders. We are in the pole position before the data, and we believe we're now even in a stronger position with respect to both first and best in class. Now let's get into the details. Dr. Liu?
Thank you, Mark. Good morning. Before I present this set of very exciting data, I would like to take a moment to acknowledge the tremendous contributions from our internal and external colleagues who have worked tirelessly to make this a reality. The site personnel, and most importantly, our patients who participated in this study, I am presenting this on behalf of all of you. To start with, I'm very pleased to report positive results from the phase 2 ARTEMIS ulcerative colitis study, which has successfully demonstrated compelling results from PRA023 as an induction therapy in patients with moderately to severely active ulcerative colitis. The primary and all ranked secondary endpoints for cohort 1 were met in a refractory population with very severe disease, as Mark has indicated. More specifically, 47% of the subjects have previously exposed to at least one advanced therapy, which includes approved biologics, JAK inhibitors, or S1P. Over 70% of subjects per centrally read endoscopy came into the study with the highest Mayo endoscopy score of 3. The primary endpoint was met with 26.5% of subjects in the PRA023 group achieving clinical remission, compared to 1.5% of subjects in the placebo group, for a delta of 25%. The first ranked secondary endpoint was endoscopic improvement, defined as endoscopy score of 0 or 1. It was achieved by 36.8% of subjects in the PRA023 group, compared to 6% in the placebo group, for an effect size of 31%. Immunogenicity rate was low at 10.3% and did not impact efficacy or safety. And as Mark has mentioned, an interim analysis for the CDX signal in cohort 1 indicated positive trend for enhanced efficacy, treatment effect in companion diagnostic positive subjects. And lastly, PRA023 was well tolerated with no safety signal identified. Just to remind you of the study design, the phase 2 ulcerative colitis study was designed with 2 shots on goal. The placebo-controlled cohort 1 was designed as a potentially registrational study to demonstrate the efficacy of PRA023. Cohort 2, the box at the bottom, the companion diagnostic extension cohort, is intended to assess the effectiveness of the companion diagnostic. Both cohorts have the same inclusion and exclusion criteria, with the exception being cohort 2 includes only companion diagnostic positive subjects, and that cohort is currently enrolling. The study enrolled moderately to severely active UC subjects who have failed at least one conventional or approved advanced therapy. The primary endpoint was clinical remission, as defined by the modified 3-component Mayo score at week 12. All secondary endpoints as listed on this slide here were ranked and alpha controlled. Patients were stratified by their companion diagnostic status and prior advanced therapy exposure and randomized to PRA023 or placebo. The CDX status was kept blinded throughout the study. At the completion of cohort 1, when all cohort 1 subjects have reached week 12 or discontinued from the study, we locked the database and conducted an interim analysis in the CDX positive subgroup. Dr. Laurent will present the interim CDX subgroup data in subsequent slides. We randomized a total of 135 subjects into cohort 1 of this study. While 10% of the 67 subjects discontinued from the placebo treatment group for reasons outlined in the box on the left, none of the 68 subjects discontinued from the PRA023 treatment group. This slide shows the baseline demographics and baseline disease characteristics. The first thing to note is that the placebo and PRA023 treatment groups are quite well balanced, with the exception of maybe slightly higher disease severity in the placebo group, as you can see highlighted, with 79% of subjects with the highest endoscopy score of 3. And on that note, I should point out that the overall population, as Mark has mentioned, is one of the most severe patient populations studied, with over 70% of subjects with endoscopy subscore of 3. Furthermore, approximately a third of subjects have previously used 2 or more advanced therapies. As mentioned in the summary slide, the effect size of the primary endpoint of clinical remission was 25%. For the first secondary endpoint of endoscopic improvement, the delta was 31%. Remember that this is in the context of 70% of patients starting with an endoscopy subscore of 3, needing to go down to 0 or 1 to meet this endpoint. The next secondary endpoint of clinical response effect size was 44%. All other cohort 1 secondary endpoints were ranked and controlled for multiplicity. All endpoints achieved statistical significance, including symptomatic remission.
So there's no confidence in our precision medicine approach, and to maintain momentum towards phase 3, we decided to conduct an interim analysis of the ARTEMIS-UC cohort 1. This essentially allowed us to take a look at CDX performance several months ahead of schedule, even if, as a result, the analysis could only be performed on a limited number of patients. Only 32 CDX positive patients at cohort 1 wasn't powered the statistical analysis of CDX selection. Nevertheless, response data from the subset of CDX positive patients in cohort 1 demonstrated a placebo-adjusted clinical remission rate of 37.5% compared to a placebo-adjusted remission rate of 25% for all comers. This difference exceeds the 10% increase defined as meaningful by payers and physicians in our own market research, and is remarkable given how high clinical remission already was in all comers. 12% improvement on top of an already strong clinical data set represent a very encouraging trend for what is still the first ever prospective use of a companion diagnostic in an IBD clinical trial. We are enrolling the ARTEMIS-UC expansion cohort 2, to further evaluate the performance of the CDX in a larger number of patients, with results expected in the second quarter of 2023.
I apologize for the audio being cut out. You would go back one, thank you. Treatment with PRA023 led to statistically significant decrease in symptoms as assessed by partial Mayo score as early as week 1, as you can see here. The effect persisted throughout the induction period. Turning to our safety data. PRA023 was well tolerated as an induction therapy. On the PRA023 treatment group, there were no treatment emergent severe adverse events, AEs leading to discontinuation, SAEs, or infusion reaction. Infection rates were similar across the treatment groups, as were adverse events assessed by the investigator as related to study drug. This slide lists all treatment emergent adverse events that occurred in more than 2 subjects in either treatment group. And as you can see, there were no differences in frequency of occurrence between the 2 treatment groups. So in summary, the ARTEMIS-UC study demonstrated a highly differential profile with strong efficacy associated with favorable tolerability. We also observed favorable signal in the CDX positive subgroup in the interim analysis conducted, and are keen to complete the recruitment of cohort 2 and share the complete set of data with you before the end of second quarter in next year. Turning to Apollo-CD, it's a phase 2A study. We also demonstrated positive results that constitute robust proof of concept. The population was highly refractory, with 31% of subjects who have previously been exposed to a biologic therapy. The endoscopic response rate was 26%, and the clinical remission rate was 49%. Both were statistically significant compared to the historical placebo rates of 12% for endoscopic response and 16% for clinical remission. Endoscopic response and clinical remission were also observed in subjects previously exposed to biologic therapy, and that's 71% of the subjects, as well as with or without concomitant therapies in corticosteroid or immunomodulators. We observed an early onset of action as measured by both decrease in symptom scores and disease biomarkers. The rate of immunogenicity was also low at 14.5% and did not impact efficacy or safety. TRA-023 was well tolerated with no safety signal identified. To remind you of the study design, this was an open label study with inclusion-exclusion criteria typical of a study enrolling subjects with moderately to severely active Crohn's disease. The design utilized a pre-specified placebo rate based on historical data to assess efficacy for the primary endpoint of centrally read endoscopy response and the first secondary endpoint of clinical remission at week 12. 55 subjects were treated in the study. Similar to the UC study, the completion rate was extremely high at 96% in the 55 subjects that were treated. One patient discontinued due to adverse event of Crohn's disease exacerbation, and one discontinued due to lack of efficacy. This slide displays the baseline disease characteristics and demographics. I note that 60% of subjects were from North America. The mean disease duration was at over 10 years. More than 50% of subjects were previously exposed to 2 or more biologics. More than a third of the subjects were previously exposed to 3 or more biologics. TRA-023 achieved significant differences for the primary endpoint of endoscopic response as defined by decreasing endoscopy score of 50%, and secondary endpoint of clinical remission defined as a CDAI score of less than or equal to 150 points. Similar to observations in the ulcerative colitis study, TRA-023 also significantly reduced signs and symptoms of disease as measured by change in CDAI score as early as week 1. TRA-023 was also well tolerated in the Crohn's disease population with no treatment emergent SAEs, severe adverse events leading to discontinuation, or infusion reaction. The 45.5% infection rate is not uncharacteristic of a study in this population. And if you look at the list of adverse events that occurred in more than 2 subjects in this study, you will see that these are all common adverse events. I will now turn the podium back to Dr. Laurent to present the very exciting companion diagnostic results from this study, followed by some cutting-edge translational science from the biopsy and serum samples from the Apollo-CD study.
Thank you. Now, in Apollo-CD, we confirmed that our pre-specified genetic markers can be used to identify a patient population with increased response to TRA-023. Also, our original algorithm provided only limited benefit in clinical responses a more recent Crohn-specific algorithm that uses the same genetic markers demonstrated enhanced performance across both clinical and endoscopic outcomes. This alternate Crohn-specific algorithm, which was not trained on clinical data, increased endoscopic response from 26% in all comer to 45% in CDX-positive patients, a 19% absolute delta. As a result, we plan to advance this alternate Crohn-specific algorithm with PRO2/3 in registrational studies for Crohn's disease. Before we move to fibrosis and inflammation biomarkers, I just would like to briefly summarize the findings of APOLLO-CD. As presented by my colleague, Dr. Liu, and consistently with efficacy in ARTEMIS-UC, PRO2/3 elicited a strong clinical response in Crohn's disease patients while being well tolerated and displaying low level of immunogenicity. And really, given these remarkable responses in both clinical studies, it would be logical to also expect profound underlying changes in molecular markers of inflammation and fibrosis. And as a matter of fact, this is exactly what we observed systemically and in disease tissue collected in APOLLO-CD. In Crohn's disease patients treated with PRO2/3, we observed a significant reduction in intestinal activated fibroblasts, the key players in collagen synthesis and deposition in tissue. We also observed a significant reduction in a 24-gene signature, which has been linked to penetrating fibrosis in the literature. For context, fibrosis remains one of the highest unmet needs for IBD patients, especially Crohn's disease patients, where overt fibrosis of the small intestine leads to structuring disease and recurring surgical intervention, something that isn't directly addressed by any approved IBD therapy. But the effect of PRO2/3 wasn't limited to fibrosis, and we also observed a consistent and comprehensive decrease in inflammation markers after treatment by PRO2/3. This included a reduction in circulating pro-inflammatory cytokines. As a matter of fact, a very consistent reduction across a large panel of inflammatory cytokines. Similarly, Th1 and Th17 tissue signatures were decreased in patients treated with PRO2/3, as were the numbers of infiltrating T cells in intestinal lesions. But going back to fibrosis, and again emphasizing that blocking TL1A is the first mechanism of action with a direct impact on tissue fibrosis. You can see on this slide that PRO2/3 significantly decreased the 24-gene signature that has been linked in the literature to penetrating intestinal fibrosis. As you would expect, the signature includes several types of collagens and extracellular proteases with documented direct role in tissue damage and fibrotic remodeling. We even observed a meaningful decrease in COL1A1 expression after treatment. Maybe most strikingly, PRO2/3 led to a decrease of activated fibroblasts in disease tissue. You can see that on the right side of the slide. Something that we have identified with the convolution of bulk RNA, but really demonstrating that blocking TL1A can lead to beneficial stromal tissue remodeling. It is equally striking to see how comprehensively PRO2/3 decreased levels of circulating cytokines in treated patients. IL-1 beta, IL-6, IL-17A, IL-22, and IL-9 were all significantly reduced in Crohn's disease patients who received PRO2/3, while the level of IL-10, an anti-inflammatory, anti-fibrotic cytokine, actually increased during the course of treatment. It's not easy to see changes in circulating cytokines in IBD patients, but it usually requires profound underlying changes in tissue. And this is also what we observed. As depicted on the right side of the slide, we observe a strong reduction in Th1 and Th17 gene signature in disease tissue, something consistent with previous publication on the mechanisms of action for TL1A, and also consistent with the observed decrease of infiltrating CD4 positive effector cells in patients' tissue. In summary, we've really confirmed the pronounced impact of PRO2/3 on molecular markers of both fibrosis and inflammation in treated patients on APOLLO-CD. This is something that is expected and consistent with the effect that PRO2/3 also had on clinical endpoints. We also confirmed the broad pleiotropic action of PRO2/3 on both immunity and fibrosis, and therefore its potential to also impact other immune-mediated diseases.
So let me close by reflecting on all we've accomplished. 2 positive studies in UC and Crohn's with highly disruptive efficacy and a clean safety profile. despite one of the most severe patient populations studied in this category. The first precision medicine result in I&I. Very promising signals across both trials. The first pleiotropic mechanism showing both anti-inflammatory and anti-fibrotic activity provides additional confidence in SSc-ILD and paves the way for new indications. We'll take the next several months to refine our registrational plans and gain feedback from the regulatory authorities. Given these impressive results in hand, we expect to have a number of attractive options to finance our registrational studies. Go more broadly with additional indications with PRA023, as well as to continue to advance our precision-guided pipeline. Clearly, the value of our SSc-ILD, CD30, and the rest of our pipeline has been enhanced based upon these findings. We are well capitalized today, and we'll take some time to assess our options. There's clearly no shortage of interest in TL1A. It's great to deliver on the promise of this unique program, and we look forward to communicating further our plans as they firm up. Finally, I would like to thank our founders at Cedars-Sinai, Dr. Targan McGovern, the late Todd Giamatti, Scott Glenn, Rolf Bernishka, our board, our leadership team, our employees, and our long-term investors who have stood by us during a choppy market, namely Eventide, RTW, Perceptive, Cormorant, and Cowen. I thank you for your support and belief in us. We couldn't have done it without you. We believe these data clearly validate the Prometheus platform and our pipeline and set us up for the future leadership in I&I more broadly. So with that, I would like to welcome Bruce Sands, Chief of GI at Mount Sinai, to join us on the line here for Q&A. While the data, I think, speaks for itself, it's always great to hear firsthand from one of the world's leading KOLs. So with that, operator, we'll open it up for questions.
As a reminder, if you'd like to ask a question at this time, please press star 11 on your telephone. Please stand by while we compile the Q&A roster. Our first question comes from the line of Yatin Suneja with Guggenheim. Your line is now open.
Thank you very much. Congratulations to the team on delivering these robust results. Two broad sets of questions for me. One is on the fibrosis side, and then I do have a question after that on the competition. So can you just touch a little bit on the fibrosis potential of this mechanism in terms of the gene signature changes that you saw in Crohn's disease? How meaningful is that signal? Like, what are the clinical implications of it? And if Dr. Sands can sort of provide his perspective on that finding. And then for the company is, given this anti-fibrotic potential, how should we think about indications beyond IBD, implications for the SSc-ILD indication, if at all? And then I have a follow-up after that.
We turn it to Dr. LeBlanc, and also we'll get Dr. Sands as well.
Sure. Thank you. We're particularly excited about the anti-fibrotic effect. As I mentioned, a 24-gene signature that includes several metalloproteases involved in tissue digestion and remodeling, several types of collagen trending together. The fact that we saw meaningful changes in individual genes, I think that requires significant effect in the study, the size of pathology, MMP3, MMP9 that is associated with intestinal fibrosis, MMP25, and even COL1A1, when the types of collagen deposed in intestinal fibrosis were all significantly reduced by treatment with ARO-3. The scope of that is very broad. You know, it's hard to find a chronic immune disease that is not associated to fibrosis of soft tissue. That in the for intestinal fibrosis in the lung, the scleroderma, we're already using PRO-23 in liver as well. And it really validates the fact that PRO-23 is more than just an anti-inflammatory drug, but also a drug that works directly on non-immune component of the tissue and very quickly on modulating fibrosis markers.
Dr. Sands, I want to provide you an opportunity to weigh in as well.
Yeah, I think it's safe to say that we don't have any agent at this point that has any effect on fibrosis. And I think the markers are rational scientifically. The changes that you're seeing, they do correlate with fibrosis, mainly the penetrating complications that we see with penetrating fibrosis that we see with Crohn's disease and stricturing disease. More importantly, I think we could also highlight that activated fibroblasts or inflammatory fibroblasts are increasingly being implicated in the pathogenesis of IBD in general, especially Crohn's. So it all makes sense to me, and of course, we don't know anything yet about what the clinical impact of PRA-023 will have on patients with fibrosis, but I expect we'll learn that over time. So this could be a very important innovation in the care of patients with Crohn's disease. And I would add that there's a growing body of evidence that fibrosis is not just a factor in Crohn's disease, but in chronic ulcerative colitis as well. That really does have a long-term impact on disease outcomes in ulcerative colitis too.
Got it. That was very helpful. Very helpful. One more question I have, maybe Mark, this is for you if you could. So given these data and the recent news that we heard from Pfizer out-licensing their antibody, how should we think about your positioning in the space, the lead you might have over the competition in the future, the rigor and the speed with which we go into phase 3 development?
Yeah, you're cutting out a bit, but I think your question was regarding the company in New Jersey that had a TL1A program. Look, let me be clear. This is the last time that we're going to comment on Pfizer. I think that over the last 2 years, we've obviously fielded a lot of questions, and rightfully so, given that they were a competitor in the space. The data that we've revealed today provides a clear verdict in terms of our path to best and first. And clearly, Pfizer's pivot to a next-generation antibody says it all. And so I think that the focus here should be on broader opportunity. And the fact is we're not just competing for share ownership of the TL1A class, but broader IBD and I&I.
Thank you.
Good question. Operator, next question.
Our next question comes from Chris Shibutani with Goldman Sachs. Your line is now open.
Thank you very much. Good morning and congratulations, Mark and team, on this data. 2 questions, if I may. Can you comment on the question of induction efficacy versus maintenance? What do you think about the applicability of this anti-TL1A mechanism in those 2 different settings? And remind us when we're actually going to see some initial maintenance data. And then on the companion diagnostics, you do mention that you had an adjustment to the algorithm. Can you just explain how the algorithms differ and how did it develop? Was it on post-hoc analysis? And what gives you confidence of the predictive value of this adapted algorithm for phase 3? Thank you.
Alison, why don't you take the first part, and then we'll have Olivier jump in on CDX.
Hi, Chris. Good question. So in terms of maintenance, I think we have a couple things working for us. One is the fact that immunogenicity was low and it didn't impact efficacy or safety. And 2 is the mechanism of action being both anti-inflammatory and anti-fibrotic. We're hopeful that it confers positive effect, even better effect during long-term maintenance therapy. And the last piece is just wanted to remind the audience that we actually have the subcutaneous auto-injector on hand to take it into our phase 3 studies. We do have some open label data on hand, which will be the subject of further presentations in future medical meetings.
Thanks. Olivier?
Sure. Regarding the Crohn-specific algorithm, we're very confident with the work we've done for 2 main reasons. The first one is that we still use the same genetics as the CDX that we still use in ulcerative colitis. Those are variants that we've identified a long time ago that were prospectively specified into the study. In addition, we did not use the clinical data to train the algorithm, which means we didn't feed it to the clinical data. We observed its performance when the clinical data became available. At the end of the day, it probably speaks about differences in biology between Crohn and ulcerative colitis. Keep in mind that part of her genetic signature is outside of the TL1A locus. We look at multiple pathways supporting TL1A. It's not surprising that you have to arrange your parameters with different coefficients to get the best response, but you keep really the underlying principle of buccal swab, the same genetic markers, and pre-specified scientific work supporting the model.
Great questions. Thanks, Chris. Operator, next question.
Our next question comes from the line of Chris Howerton with Jefferies. Your line is now open.
Hey, I don't know if you can hear me. The line kind of cut out. Hey, great. So first of all, congratulations on the stellar data. Truly fantastic. I guess in light of those really excellent data that we're seeing today, I guess Mark and I guess I would love to hear Dr. Sands. What would be the design features of a phase 3 registrational program to really maximize your ability to utilize this product across the spectrum of IBD patients? Like for example, how can you maximize the opportunity in moderate disease? And then a follow-up question that I had in relation to Chris's previous question on the companion diagnostic. I guess, Olivia, could you talk to us a little bit about how you're thinking about utilizing your understanding of the companion diagnostic across other indications outside of IBD and SSC? What are the things that you're thinking about to maximize your knowledge and utilization of that diagnostic moving forward? Thank you.
Yeah, great questions. Thanks, Chris. Alison, why don't you talk about our phase 3 strategy and some of the planning, and I'd love to get Bruce's thoughts as well.
Yep. Hi, Chris. So our phase 3 planning is well underway. Of course, we plan to go to the FDA and European health authorities to get the final agreement on the study design. What we do know is that PRA023 has a very long half-life of 19 days. So we will probably design the study to leverage that fact. And we will be disclosing the study design after it's been finalized.
Yeah. Bruce, would you like to comment?
Yeah, without going into details about what Alison is planning, I can just say that, typically we do, of course, explore a whole range of disease activity, and I would add severity. Activity is really the snapshot of how symptomatic the patient is at the moment that they're being seen. The severity is really the patient journey from beginning to end. And most of our studies really incorporate a broad range of patient experiences, patients with different experiences into the population. So we'll see people at the lower end of disease activity in terms of CDAI and then in terms of endoscopic severity all the way to fairly severe. And that's just very typical of our studies. It is more challenging to reach into mild disease simply because there's a lot of deviation to the mean, regression to the mean of those patients, meaning a high placebo response effect. So personally, I wouldn't want to see that mild population incorporated into a pivotal trial. Although if the safety holds up, that's not out of the question either.
Great. And then regarding the CDX and applicability in other TAs?
Yes, as you know, we're already using PRA023 in scleroderma-associated IBD, and also we're not selecting patients based on the CDX. We're tracking their status based on the genetics we have identified for TL1A. Also, the data set were small for UC and CD. What you've seen twice now is that our genetic can really move the needle into the right direction. So we're really convinced that what we're looking at is connected to TL1A, and it's a very good sign in terms of predicting TL1A importance for other indications.
Well said. Thanks, Chris, for the question. Operator, next question.
Our next question comes from the line of Thomas Smith with SVB Securities. Your line is now open.
Hey guys, good morning. Thanks for taking our questions, and let me add my congrats on the really excellent data here. A few questions. Maybe we just start with a clarifying question on the baseline characteristics. You described the previous treatments here by number of prior advanced therapies, but I'm curious if you've looked at the split of patients treated with biologics versus JAK inhibitors versus S1Ps, and if there are any differences in these populations. And then secondly, with respect to the interim CDX analysis in the UC study, you just talk about whether or not this changes any of the powering assumptions for the biomarker enriched cohort 2 portion. I think you previously talked about this being 80% powered, assuming a 30% or so clinical remission rate versus 5% placebo rate, but just curious how this interim impacts the cohort 2 readout. And then maybe just lastly for Dr. Sands, I was just wondering if you could help put the initial companion diagnostic data in perspective. How do you think about the strength of the data? How do you think about a 12.5% boost over all comers in UC? And how do you expect you'd use this in real world practice?
So Tom, thank you for the great question. In terms of baseline disease characteristics, so in the ulcerative colitis study, we mentioned that 47% of subjects were prior biologic exposed. A relatively small proportion of those were exposed to to the small molecules. Most of them were exposed to biologics, and there was no significant difference between the exposure naive and the exposed in the subgroup analyses. We will be presenting, of course, the subcuts down to the actual molecule in subsequent medical conferences. In terms of the cohort 2, as I mentioned in my presentation, I think the interim analysis was defined prior to the database lock with a very small alpha consumption. So therefore, we still have a significant amount of alpha remaining in the cohort 2. Therefore, we're not changing the design or the powering of that study. I'll turn it over to Dr. Sands to comment on the companion diagnostic in the real world.
Right. Well, first of all, I just want to underscore that this can be the very first example of a useful companion diagnostic in IBD, something we've all been waiting for. And I think prior research suggests that a 10% boost to 15% boost over all comers would be a useful increment, and I do agree with that. But I would also look at the data in another way, which is the odds ratio or relative risk, if you will, of the CDX positive over CDX negative. And if you look at that, that's an even greater exaggeration of the benefit of the companion diagnostic. And essentially, you're seeing kind of a 1.5-fold kind of boost in efficacy if you view it that way. So I think that's very important for the field. The last thing that I would say is this is such an early look. It's a relatively small number of patients. As you saw in the presentation, there are wide confidence intervals, and it may in fact turn out to be an even bigger increment. That's entirely possible within the realm of the data that's presented here. So I guess the best we can say is that this is very encouraging. Of course, we want to see the full data set before we make any full evaluation of it.
Okay, great.
Well
That's
said,
super
Dr.
helpful.
Well said. Real quick, if I could just jump in. Look, the interim look provides us an advanced, look in the future. And I'd say it bolsters our belief in our precision medicine strategy. One important data point here is that our CDX negative population in the UC study showed higher remission rates than most of the drugs that are on the market today. So I think we just need to take a step back and say, first of all, when you look at 25% all-comer remission, it is a high watermark in the class when you couple that with safety. This is Rinvoq-like efficacy with Entyvio-like safety. I don't know if there's another way to say that. It far exceeded our expectations, and I know all of your expectations. With regard to the CDX, we saw 2 very promising signals. To Dr. Sands' point, it's early, but it's very encouraging, and we have a lot of confidence that we can be the first to pioneer precision medicine, and we believe that we're the best hope to bring precision medicine into into IBD and broader I&I. So thank you for your comments, Dr. Sands. Next question, please.
Our next question comes from the line of Julian Harrison with BTIG. Your line is now open.
Hi, good morning. Thank you for taking my questions, and let me add my congratulations on these powerful results this morning. First, you have a novel mechanism here paired with remarkably clean safety. So with that in mind, it seems like there are probably many promising opportunities to assess PRA023 in combination with other targeted therapies. I guess the question is, how much do you want to prioritize that going forward, considering the monotherapy efficacy looks very good already in both indications?
Yeah, let me let Allison jump in with regard to combination opportunities first on the clinical side, and then I'm going to ask Mark Stenhouse, our Chief Operating Officer, to opine on the commercial opportunity.
Good morning, Julian. Thank you. So combination therapy, the first thing you think about is, to your point, a clean safety profile that would enable even thinking about combination therapy. And you're right, we're clearly there in terms of whether adding another advanced therapy to PRA023 can achieve even better results. I think that's absolutely something that should be explored. And I'll let Mark Stenhouse comment on the commercial side.
Yeah, and first thing I would just say is, as has been mentioned, our confidence in best in category data that's been presented today. We have always said that there's three ways to disrupt the I&I space. First is the genetic selection of targets. Second would be using a precision approach to bring upsized efficacy to a subpopulation. And third, to your point, is rational combination therapy. I think with the data we have, the unique blend of efficacy and safety, we are in a position to do all three.
Well said. Next question, please.
Our next question comes from the line of Derek Archila with Wells Fargo. Your line is now open.
Morning, Derek.
Oh, hey, good morning. Sorry, cut out there. Well, let me also offer my congratulations on the data. Very excellent. So just a few questions from us. So wanted to get your thoughts on why you saw consistent efficacy of both across the naive and the experienced population. That isn't typically the case in UC trials, so just want to understand if there's a hypothesis around that. And then also, given the severe population you enrolled, again, most of them being endoscopic score 3, I guess why weren't there more biologic experienced patients? So that's kind of the question one. And then question two is, and Mark, maybe you could just expand on some of the comments that you just made and maybe even get some color from Dr. Sands. So given the data that you're seeing with the companion diagnostic and in all comers, I guess how do you think about the commercial positioning within UC now, both in, I guess, maybe, front line and later line? And would you assume that similarly for future indications also, given the data that you've seen?
So let me start with the naive experienced question. So I think the effect size that you're seeing here is remarkable. And in that setting, whether you subset it into bio-naive or bio-failure, they all cross statistical significance. Numerically, there is a small difference, and you're right. The bio-failure or the bio-experience do have slightly lower efficacy, but they are all statistically significant. Then to your question of then why aren't they more refractory? So when you talk about the disease severity, it doesn't necessarily jive with the refractoriness of it. And you're right in pointing out that our population is indeed very severe, but in terms of refractoriness, only 47% of the patients were bio-experienced. But I should note that of the 47%, significant proportion were experienced with 2 or more prior biologics.
To Allison's point, I think that the data across both the all-comer as well as the trend that we see in companion diagnostic is extremely compelling. And we said all along that our trial design provides multiple ways to win. We'll be honest, going into this, we kind of set expectations at 10 to 15% because that's kind of where the market was. And clearly delivering 25% placebo-adjusted remission is, best in category. Our label strategy all along has been to be able to access that have universal access to the moderate to severe UC and Crohn's population. And then within the CDX population, we wanted to be able to, the positive population, be able to negotiate for frontline therapy with biosimilar. I'll be honest with you, I think there's actually two ways to win here based upon these outcomes. One is you could argue that 25% placebo remission, if replicated in the phase 3, would put you in a very strong position to compete for frontline business with biosimilar. Similarly, being able to show a 10 to 15% absolute improvement with a companion diagnostic would further refine that case and reduce gross to net discounts on the commercial setting. So I think that the aperture has been opened. The expectations in terms of remission have been exceeded. And I think we're in a great position to continue on this journey of multiple ways to win. Bruce, any thoughts?
If Dr. Stan add anything to that, that would be excellent.
Yeah, no, I completely agree with what Allison and Mark just said. I mean, if you look at the field, sure, we do have biosimilar anti-TNFs, more and more of them. They really come with the baggage of safety. We know their efficacy. The efficacy is good, and we use them across the spectrum of IBD, moderate to severe. But really, it's the safety, and we know that it's not just the safety of the monotherapy, because we know the better efficacy is with combination therapy. So we have the baggage from the combination as well. So I think when you see superior efficacy and you see superior safety, this is going to win the day. And there's no question it could be first line. UPA is not going to be first line unless the FDA changes their stance. The S1Ps, sure, they can be used as first line advanced therapy, but they don't show nearly the efficacy that we're seeing here. And they do have some safety issues, manageable, I think, but still those issues do exist. And I think those are the things that have impeded uptake in the market of this class of agent. I think there's really great promise here.
Excellent. Thanks so much for the color, and congratulations again on the data.
Thanks, Derek. Great questions. Yasmin, you're next. Thank you for the question.
Thank you so much. First of all, really congratulations to the stellar data, and more importantly, to an impeccable execution. Really reporting, a 1.5% placebo rate is really outstanding. Goes back to how well done the study was done. So really wanted to start off with that. And then a couple questions, maybe if you could comment on where you are in terms of your sub-Q formulation, how soon, bioequivalence to the infusion, how soon would be ready for phase 3. And then secondly, I think also you guys have done a really nice job on warehousing patients for phase 3 as you have been working on our diagnostic kit. So if you could just give us a quick cut on how many patients you have sort of ready to go for phase 3 across UC and Crohn's. And then the last question is for Allison. Did you look at whether there was any differences in efficacy between patients in Northern America versus Eastern Europe? And thank you again for taking my questions.
Hi, yes. Thank you for your really nice congratulatory note. So your first question, sub-Q formulation, we have completed the bioavailability study in normal healthy volunteer, and we found more than 80% bioavailability. So the auto-injector is ready to our phase 3 study, which can start next. In terms of your comments on Enroll 360, that's a platform where we have global study-ready sites. That study has enrolled more than 2,000 patients that we can draw on in subsequent studies. And we are currently continuing to enroll cohort 2 of the ARTEMIS-UC study using that platform. The differential effect of North America, so I should note that in the study, 60% of the patients came from North America. In the UC study, approximately from North America. We will continue to entire data set analysis that hasn't done. I believe that is significant. The geographic distribution. Great question.
Great questions. Yeah, as usual. Thank you so much.
Thank you.
Next question, please. You have enough for two more questions.
Our next question comes from the line of Tiago Fauth with Credit Suisse. Your line is now open.
Hi, congrats on all the updates. Really stellar results here. Just a couple for us. Gating steps for the registration program in UC. I'm curious if cohort 2 data is actually a potential gating step or not. And it cut off a little on the last reply, but I'm curious about the bridging strategy and any additional dose exploration trials, either in patients or healthy volunteers that you intend to do, or is that kind of going to be folded into the registrational program? Thanks.
Alison, you want to hit the bridging?
I'll hit the bridging, and I didn't hear the first.
The first question was around is in cohort 2 a gating item to begin the registrational study?
Right. So let me hit bridging first. Yes, so we have completed bridging in normal healthy volunteer for the subcutaneous formulation. We do not need to do further work to initiate the phase 3 study with our auto-injector. In terms of whether cohort 2 is a gating item, it's not. The way the phase 3 will be designed, and we have not disclosed the phase 3 design yet, but Mark has mentioned that the labeling strategy is to obtain a broad indication with differential efficacy in companion diagnostic patients. So therefore, we will be moving forward with all of the regulatory discussions and prepare for the initiation of phase 3 without waiting for the cohort 2 results.
Thank you, Alison.
Got it.
Operator, we got 2 more, but only 2 minutes, so let's go.
Our next question comes from the line of Gregory Renza with RBC Capital Markets.
Great. Good morning, Mark. Good morning. Congratulations, Mark and team, on the excellent data. Maybe just 2 quick ones from me, if I may. Mark, you commented just earlier on replicability. I was wondering if we could just ask Dr. Sands to comment on his views on data replicability when it comes to clinical remission in UC, and how Dr. Sands, you sort of think about the absolute and relative clinical remission and that importance to you. And then lastly, Mark, just for the team, I just wanted to confirm, or if in case I missed it, whether the ADAs, whether you saw any impact on pharmacokinetics in those patients who had ADAs, and just your level of confidence that those ADAs, and certainly while small, that they can be managed and not an issue moving forward. Thanks so much, and congrats again.
Yeah, fabulous questions. Dr. Sands.
Sure. If I understood the question, you're asking about what I think about the replicability of these outcomes and the significance of them. I think you have to understand that we have the prior, at least, of the Pfizer data in the class, suggesting that this agent, or this approach, this target, is an effective target. It is an important target. We saw estimates from the Pfizer study that are not dissimilar to what we see here. But here we see a very robust study, very well executed, as you can see, and very much in line with what we've seen with upadacitinib studies. So my full anticipation is this will replicate. I think the outcomes speak for themselves. These are standard outcomes that we've looked at, the endpoints of clinical remission, endoscopic improvement, and all the other things that you saw, all the secondary outcomes are all in line with each other. And it's the concordance of the outcomes that really gives me confidence in the replicability. I think clinical remission is really important in UC because it is the composite in the same patient of both patient-reported outcomes and the endoscopic outcome, which is objective.
Thank you, Dr. Sands. So as to your question about whether ADA impacted PK, I would just say that ADA did not impact safety. In fact, we did not see any infusion reaction, and it didn't impact efficacy because we did break out the subgroup of the patients, the low proportion of patients in whom ADA was observed, and there was no difference in efficacy in this subgroup. We have not yet completed our PK analyses since this was just the top-line results, but we will complete it and report it in future medical conferences.
So with that, that wraps up all the questions. To say we're enthusiastic is the understatement. We're so proud of the team. We appreciate the support, and we're very excited to advance this therapy in IBD in phase 3 studies, as well as other new indications that we'll talk about next year, as well as advancing our pipeline. As I mentioned, clearly this adds a lot more confidence to our precision approach, to SSC-ILD, as well as the future pipeline, CD30, and other targets to be announced. So with that, we'll wrap up and look forward to seeing you guys at JP Morgan.
This concludes today's conference call. Thank you for participating. You may now disconnect.